My name is Nick Seyfried. I'm an associate professor in the departments of Biochemistry and Neurology at Emory School of Medicine. My research is focused on the use of quantitative mass spectrometry techniques and proteomic technologies to characterize Alzheimer's disease and related neurodegenerative diseases, human brain tissues, and the modifications of the proteins within these tissues, and how these relate to disease pathogenesis. And then ultimately what we want to do is take the signatures out of the human brain and to translate these into biomarkers for the early detection and diagnosis of Alzheimer's disease and related neurodegenerative diseases. We use complementary label-free and isobaric tandem mass tag approaches to quantify proteins and these complexes and these modifications in disease. One of the things that is a limitation to label-free based approaches. In general, in a 90-minute to an hour and a half, 2-hour gradient, we're only seeing about 3,000 to 4,000 proteins. And if you run multiple samples, you will have some chromatographic drift and some signal intensities that depreciate over the number of samples you run. And this leads to more missing values. Whereas with isobaric tagging using the tandem mass tag approaches, we've been able to label a pooled standard that allows us to control for quantitation by batch over a number of samples. And the other thing it lets us to do is to deeply fractionate the peptidomes from multiple samples simultaneously. And this gives us enormous depth of the proteome, which we like because we can now go from, instead of 3,000 proteins from an individual, to about 8,000 to 10,000 proteins from a single individual sample. And now we can do this over hundreds of samples. And with these pooled standards that you dedicate one of your TMT channels for, you can better control for batch drift and signal intensity across samples. Because we're looking for as many individual proteomes as possible. The ability to do 10 samples simultaneously, or at least 9 when you have one of the channels dedicated to a standard, allows us to get at the power that we'll need to look at individual trajectories of proteomic changes in biofluids collected from living patients. So I think as we move forward and as the multiplexing technologies advance, it's just going to dramatically increase our throughput in terms of individual proteomes that we're going to be able to analyze in a clinical setting, which will have major impact on biomarker discovery and validation.